My name is Dr. Ibrahim Ismail of the Department of Biochemistry, Federal University, Dutse, Nigeria. My research interest is finding a new therapeutic target for the management of diabetes mellitus. In the course of our investigation, we have come across an enzyme that plays a vital role in the development of diabetes mellitus, and that enzyme is carbonic anhydrase. Carbonic anhydrase is a ubiquitous zinc metalloenzyme with molecular weight of 30 kilodalton that is found in almost all organisms. Carbonic anhydrase is one of the fastest enzymes known, with catalytic rate of approximately 1 × 10 to the power 6 per second. That is, each enzyme molecule can hydrate 1 million molecules of carbon dioxide per second. Carbonic anhydrase is involved in many biosynthetic reactions under both physiological and pathological conditions. It is also involved in pH regulation, carbon dioxide and bicarbonate transport, ion transport, and bond resorption. Carbonic anhydrase provides bicarbonate for the synthesis of fatty acids, urea, and pyrimidine. The enzyme plays a very important role in hepatic glucose production by providing bicarbonate as a substrate for pyruvate carboxylase reaction. Carbonic anhydrase utilizes the end products of both aerobic and anaerobic glycolysis by recycling them and making them available for more glucose synthesis, thus creating a continuous cycle via both the aerobic and anaerobic pathways that keeps generating more energy in the form of ATP. Under condition of exclusively anaerobic glycolysis, lactate is produced as the end product, which is removed by carbonic anhydrase facilitated transport via the monocarboxylate transporters from glycolytic cells such as muscles and red blood cells, to be taken up by the liver via same carbonic anhydrase facilitated lactate uptake for hepatic glucose production. Whereas, under condition of aerobic glycolysis, the end product, carbon dioxide, is converted to bicarbonate, which has 3 phases. Number 1, used as buffer. Secondly, used as ion transport. And thirdly, used as substrate for pyruvate carboxylase for hepatic glucose production. Both bicarbonate and lactate are necessary for continuous hepatic glucose production. Lactate, alanine, and other glucogenic amino acids alone cannot drive gluconeogenesis without carbonic anhydrase-derived bicarbonate. Therefore, inhibition of carbonic anhydrase will affect hepatic glucose synthesis and thus overall glucose homeostasis. Carbonic anhydrase are widely used in clinical practice as diuretics and antihypertensive drugs. In one of our studies with streptozotocin-induced diabetic rats, we found that inhibition of carbonic anhydrase significantly decreased hyperglycemia and improved antioxidant defense system. Recent studies showed that carbonic anhydrase inhibitors may provide a novel therapy for obesity, diabetes, cancer, infection, and Alzheimer's disease. Therefore, development of new therapeutic compounds that can selectively target hepatic carbonic anhydrase are essentially needed in designing new medication that can reduce hepatic glucose production for the effective management of diabetes mellitus.